Thank you so much for the opportunity to speak and to present my work to you. So today I'm going to talk to you about some omics approaches to identify functionally potent umbilical cord blood units. So it's probably no news to this audience that when we talk about hematopoietic cell therapies, these therapies really require functionally potent hematopoietic cells. So hematopoietic cells can look like hematopoietic stem and progenitor cells, but unless they actually have that functional potency, they're not very useful to us. And so in practice, what we actually see is that a lot of these cells can be cleared. They don't want to stick around. They don't want to reconstitute the unablated hematopoietic system. And so this is a quote actually that was co-published with my friend here, Wouter, where we talked about the need for selecting potent units and how this is going to evolve as there become more applications. And this is not something that we came up with ourselves. This is a bell that's been rung quite a bit by a lot of people in this room over the years. So why does understanding what drives potency matters? So it can improve the selection of donor units without the need for treatment. So this is a really nice graph here by Dr. Cohen's group that shows how they were able to expand that square of usable units using treatment of UM171. And we're hoping maybe if we can select more potent units, we can expand that square, again, just through the selection of units. Can we rescue those units that fall below these acceptable CD34 and TNC doses? Additionally, we might be able to engineer more potent donor units. So if we know what drives potency, can we actually edit these cells to become more potent? Can we maintain potency for cell therapies that require ex vivo manipulation? Gene editing, if we're going to develop CAR cell therapies from stem and progenitor cells, these require ex vivo manipulation, but we know that as we grow these cells ex vivo, they lose their potency. They become distinctly different over time in culture. And then again, can we apply these approaches to novel therapies? So what we hope for here is to use these omics approaches not just for transplantation, but potentially as a platform to improve other therapies as well. So this is sort of the approach that I take. It starts with this sort of potent omics screen, and I'm not going to list everything on this, but hopefully the idea is that we can sort of filter this down into relevant targets that will actually be used to improve hematopoietic cell therapies. And today I'm going to give you just a little bit of a taste of what we've been working on and talk specifically about 3 different screens that I've been performing. So the first one is this transcriptomics screen where we're looking at the RNA sequencing profile of cord blood units with known engraftment potential in mice. So we're using NSG mice for that. The second one is where we're actually sequencing cells before and after a homing period, again in a mouse model, using those NSG mice that are able to reconstitute a human hematopoietic system. And the idea here is that hopefully we can get a picture of early homing and engraftment gene signatures by looking at these cells before they go into the mouse and the cells that are actually stuck there in the bone marrow after 24 hours. And then finally, we're using the third screen here is actually using a method that we know improves hematopoietic stem and progenitor cells to see what exactly is improving within those cells, and can we get a picture of what is driving the potency within that improvement. And so again, please come by my poster later. I'd love to talk about this all night, but I've only got a few minutes, so I'm just going to give you a couple of quick images on what we did here. So this first one was looking at that transcriptomic screen 1, where we're looking at engraftment in mice and whether that can tell us something about potency-associated gene signatures. This is an example of flow cytometry plots that show you what would be a low engrafting mouse and a high engrafting mouse. And we have the sequencing data from those cord blood units prior to the cells being transplanted. We found a lot of gene expression differences between these two. This is an MA plot showing an overall view of gene expression differences. And one of the things that popped out among numerous targets was this program associated with dehydrogenase activity, which if you're familiar with Dr. Kurtzberg's potency assay was something we were excited to see because it's kind of a proof of concept. It shows the things that we know should pop out associated with potency are indeed popping out in this assessment. The next assay we did was this, where we were transplanting these NSG mice with CD34 cells, and we were sequencing the cells before and after these cells had the ability to home to the bone marrow. I'm showing here just a representative FACS plot showing that we were able to isolate human hematopoietic cells, and just to confirm that we are actually sequencing the human hematopoietic cells and not the mouse recipients, These are the reads that are associated with the human genome versus the mouse genome. You can see we were obviously sequencing the human cells, and so we're very confident that the signature is actually associated with those human hematopoietic cells. And what we found actually was kind of surprising. My initial goal with this was to find a homing-specific signature. It seems like it's really more associated with that early engraftment, though. We missed the homing window. It should have been potentially earlier, and we've got a couple of new approaches that we're using for that hopefully I'll be able to talk to you about next year. But one of the gene programs that popped out from this were proliferation-associated genes, and here's just a representative list of genes that were either entirely lost in that early engrafted cell population or that were completely newly expressed genes in these cells in that early engrafted population. And then finally, that one of the things that came from this, from these two profiling assessments were that gene programs associated with oxygen dependency actually popped out in these and a couple of other transcriptomic screens that we did as well. And so this kind of makes a very long story short, but we've been pursuing this as a separate project in the lab, looking to see whether if we change the oxygen tensions that these cells are grown in, whether it affects the potency of these cells. This is just one representative slide showing that if you expand these cells in different oxygen tensions, you get different amounts of expansion of total nucleated cells. And even though you get probably a 20-fold higher increase in expansion in higher oxygen tensions, you actually get better engraftment in mice in the lower oxygen tensions. It suggests that the amount of functionally potent cells within those low oxygen tensions is actually much more dense there. And I just want to mention that a really talented grad student out of the Capitano lab, Sarah Gutsch, is presenting this work in a poster, and you should definitely go find her and talk to her about it. And so, sorry, this next one is a big image, so it takes a while to pop up. There we go. And so what we did, using that information, knowing that these cells are much more highly dense for potent cells, is we did a single cell profiling of cells grown at different oxygen tensions. So what you see here is a UMAP plot showing the single cell profile. This includes input cells, so cells that were not manipulated at all, cells that were expanded in 5 different oxygen tensions for 2 days, and cells that were expanded in 5 different oxygen tensions for 7 days. And then we were able to manually annotate these for all these different cell populations you see here. Again, I'd love to talk more about this with you, but just for a taste of what we're looking at here, what you can see is actually that the cells grown at 1%, the low oxygen tension had a much higher percentage of these sort of unexpanded HSCs and MPPs, what we consider these potent HSCs and MPPs. And a lot of genes actually have popped out as being interesting in this. One of the ones that I'm particularly interested in is PRSS2 gene. That's very highly oxygen dependent and very highly enriched in these potent stem and progenitor cells. And then another couple of genes too, the one that I'm most interested in highlighted at the top, DDIT4, again, very highly oxygen dependent. It's associated with mitochondrial function, which we know is also very important for stem and progenitor cell potency. And to pursue this a little bit further, we then looked, do they actually predict potency? And this is using mouse models, so keep that and take that with a grain of salt. What we found was if we looked at CD34 cells overall, DDIT4 expression, those that were highly expressing actually had higher engraftment in mice compared to those that had lower expression, which had lower engraftment. And PRSS2, which I told you again was more specific for those stem cells, if we look specifically at the HSC signature, we see that HSCs expressing high levels of PRSS2 have a much higher engraftment capacity compared to those that are expressing low levels of PRSS2. And so it suggests that these models were actual ways of finding these potent genes, and we're able to validate some of those now, and we're working further on that in the lab right now. So what are the impacts of this? So we hope that these initial screens have revealed a lot of genes and gene programs associated with stem and progenitor cell function that we can serve as research interests for my lab, for Megan, my close collaborator's lab, and for a lot of other people as well. And then translationally, the happy side effect of studying what makes stem and progenitor cells tick is that we hope it has immediate translational implications. So can we use this information to develop a gene potency panel? By just extracting RNA from cord blood units, can we actually say whether these cord blood units are going to be more potent or not? And then hopefully, like I mentioned earlier, we can use this as a platform for determining potency of other units and other hematopoietic cell therapies. And we think that this is applicable regardless of donor source and target cell type. And so quickly, I just want to acknowledge my collaborators, people who are bolded or people who were actively involved in this research. I am recruiting. I know that this is probably not the crowd that's going to come and work for me, but if you know any students that are graduating soon or young postdocs who are looking for a move, please put them in touch with me. I would love to work with them. And then I will just mention one other thing. I want to just reemphasize how important cord blood is for people like me who are doing basic research. I know that's been mentioned a lot today, but I just want to say, like, none of my research is possible without these cord blood units. In my opinion, it's the absolute best source for basic hematopoietic studies. And so if you know a scientist and you can help them by providing cord blood units for them, please do so. And if I have time, I will take any questions.
I'll come back here. I guess I should go up there. Does anyone else have any questions before I? Thank you so much. That was a great talk, and thank you for your plug at the end on behalf of someone who is just like you, who needed cord blood. I'm curious if, when you showed the data of HSCs and MPPs, right, and you said you had longer-term engraftment, could you expand a little bit on what you mean by longer-term engraftment, and did you look at SRC frequency? And these MPPs, do you actually see lymphoid development later? I'm asking because I guess the basic question is, how do you know you really have an HSC and not, a true long-term stem cell versus just an intermediate?
Yeah, absolutely. So we did, when I divided by high and low engrafting, at least on what I showed here, actually was, it was SRCs that it was divided on. So it was divided on the the mean total SRCs from the entire group, of mice that we used. And so that's how we determine stem cell. We're looking actually at the entire range of engraftment kinetics though, including lymphoid engraftment. Again, that's actually the focus of Sarah's work, so I would highly recommend stopping by her poster. But yeah, we want to be able to tie these different progenitor cell populations to the engraftment kinetics. So we're actually sorting based on immunophenotype and then looking at engraftment kinetics across the entire transplantation experiment. Thank you.
In Royal Manchester Children's Hospital in UK. It's truly an honor to present our work in the Best Paper Award category, and I would like to sincerely thank the juries. Due to unforeseen circumstances, I'm not able to be available in person to present the paper and will be delivering the paper through this recorded video presentation. We know cyclosporin forms the major GVHD prophylaxis in allogenic stem cell transplantation, being a part in more than 70% of the transplant schedules. However, the benefits of GVHD prevention offered by this drug in the setting of acute leukemia transplant is offset or expected to offset by reducing the graft versus leukemia effect, thereby raising the leukemia relapse risk. Previous studies have reported a relationship between cyclosporin exposure and the risk of post-transplant relapse in leukemia. The degree of immunosuppression to prevent GVHD while keeping an adequate graft versus leukemia effect has not been established. The T-cell replete cord blood transplant might be considered the most preferred donor cell source in AML with superior GVHD-free, relapse-free survival. We report a multicentric national experience of the relationship between cyclosporin exposure and the time to relapse in a large homogeneous cohort of pediatric AML patients receiving a T-cell replete cord blood transplant adjusted to the pre-transplant MRD status and GVHD. It is A retrospective study and data was collected from 7 pediatric transplant centers in the UK on patients who have had a T-cell replete cord blood transplant for high-risk AML between 2013 and 2023 over a period of 10 years. Case records were analyzed to get patient, donor, transplant characteristics, GVHD prophylaxis, cephalosporin levels in the first 8 weeks, and the transplant outcomes including GVHD and immunosuppressive treatment used, relapse and transplant-related mortality. The primary endpoint was time to disease relapse and the relationship between this endpoint and cyclosporin area under the curve or exposure and the duration of cyclosporin adjusted to MRD and GVHD status. The secondary analysis included the relationship between time to disease relapse to the use of additional immunosuppressive treatment and its total duration. The study was investigated by multivariate Cox regression analysis. A total of 125 children had a T-cell replete cord blood transplant in the last 10 years in the enrolled centers. One patient was excluded for the lack of follow-up on relapse data. 118 patients had the data available for cyclosporin levels and the duration of cyclosporin was available on 110 kids. This is the demographics of the study population of the 125 kids enrolled in the study. The cohort included 124 patients, of whom 66, that is 53%, were MRD positive pre-transplant with a median follow-up of 737 days, and the range being 24 to 3,540 days. All were given cyclosporin and MMF as GVHD prophylaxis for a variable duration post-transplant according to each center's protocol. 52% of the group experienced severe acute GVHD and required treatment with systemic immunosuppression, which included steroids in all cases with or without second-line agents. The mortality rate was 40.7%, of which nearly 3/4 occurred in MRD positive group. Overall, relapse formed the leading cause of death, causing 68% of the total deaths in MRD positive group. It constituted only 41% of the deaths in MRD negative group, however. The treatment-related mortality was 13% and accounted for the most prominent cause of mortality in MRD negative group. The median time to relapse was 180 days and nearly a third occurred very early within 6 months. GVHD-related deaths remained very low, 9% of all-cause mortality, and was spread evenly across MRD positive and negative group. 7% of those with severe acute GVHD died secondary to GVHD-related complication. This is the relapse-free survival curve of the entire group using Kaplan-Meier method. The relapse-free survival is around 64.5% with a median follow-up of 737 days. This is the Kaplan-Meier relapse survival curve of the groups with or without severe acute GVHD in MRD positive and negative group. The green curve represents a survival curve of the MRD negative population without acute severe GVHD, closely followed by the red curve representing the survival curve of the MRD negative population with severe acute GVHD. The blue curve represents a survival curve of the MRD positive population with severe acute GVHD, and the purple one indicates the relapse-free survival curve of the MRD positive population without severe acute GVHD. What we infer from this is the relapse-free survival is greater in the MRD negative group without severe acute GVHD than in those with severe acute GVHD. However, the difference as assessed by the log rank test on Kaplan-Meier curves was not statistically significant. However, by comparison, the relapse-free survival is significantly lower in the MRD positive group without severe acute GVHD than in those with it, with a p-value of 0.034. The Cox proportional hazards model using 95% confidence interval showed a significant inverse relationship of the area under the curve of cyclosporin, that is the exposure of cyclosporin in the first 8 weeks on the time to relapse with a p-value of 0.01, and such a significant relationship maintained even after adjusting for MRD status and the presence of severe acute GVHD. There was a significant inverse relationship between the duration of cyclosporin prophylaxis and the time to relapse, with the p-value being 0.04. However, the relationship was no longer significant after adjusting for the MRD and the GVHD status. 55% had the systemic immunosuppressive treatment for severe acute GVHD. The study did not find any effect of systemic immunosuppression or the duration of immunosuppression on the time to relapse. The p-values were 0.05 and 0.2 respectively. The degree and duration of immunosuppression to prevent acute GVHD while preserving the desired GVL effect remains a holy grail, I should say. The study shows a wide range of practice variation between different centers with desired therapeutic range and the duration of cyclosporin. As one could imagine, it reflects the inadequacy of mastery in immunosuppressive prophylaxis and an unmet need in evidence-based standardization of GVHD prophylaxis. The next question that arises that is of relevance to address here is, would we be right in advocating a universal approach towards GVHD prophylaxis across all AML transplants? What we infer from the study is a one-size-fits-all approach towards GVHD prophylaxis in AML transplants is not ideal and will not help fetch the desired relapse-free survival. It is quite clear from the data that GVL is essential for cure, in particular in the highest risk group of MRD positive AML. A carefully designed GVHD prophylaxis with deliberate maintenance of lower therapeutic levels and early withdrawal of cyclosporin prophylaxis is needed to optimize the GVL effect and maximize the relapse-free survival in this risk group. In MRD negative group, however, we observed that TRM might be the major cause of mortality and severe acute GVHD might not necessarily result in better reduced relapse risk. In these cases, a more conventional approach to the duration and the therapeutic levels can be exercised. This study lacks the power to instruct the desired cutoff of therapeutic levels of cyclosporin and its duration for the two MRD groups in order to reduce the relapse risk. Further prospective studies with larger study population will be needed to confirm this observation and establish these cutoff points. Secondly, it is a common anxiety for all treating physicians while treating acute GVHD with systemic immunosuppression is whether the systemic immunosuppression might adversely affect the GVL effect and favor relapse in its own right. However, reassuringly, the secondary analysis is instructive that the addition of immunosuppression or its duration in the presence of severe acute GVHD doesn't influence the relapse risk and the relapse-free survival remains largely unchanged. To conclude, this is the largest homogeneous cohort of T-cell replete cord blood transplant in AML investigating the role of cyclosporin prophylaxis on relapse. It confirms the importance of GVL effect and its relationship to GVHD, particularly in MRD positive patients, as relapse-free survival is significantly better in those who experience severe acute GVHD. Most relapse is early, and increased cyclosporin exposure in the first 2 months of transplant reduces the time to relapse in AML and is an independent risk factor for relapse even after adjusting for MRD and GVHD status. In the presence of severe acute GVHD, the systemic immunosuppression and its duration does not result in an increased risk for relapse. This study supports, therefore, the development of novel protocols and alternative immunosuppression regimens during cord blood transplant in the highest risk MRD positive AML. I would like to thank you for patient listening and above all would like to thank everyone for the opportunity to present our work with an important message that has practical implications. I would also like to thank Professor Rob Wynn, who is my mentor, who will be available to take the questions now. Thank you.
Dr. Nguyen, are you here? If you are, make yourself known. Okay. I don't see Dr. Nguyen, so I'm sure he what? Okay, it sounds like he is not here. So let's move on to
I'm here.
Wouter's here. So Wouter is ready and poised to do our Best Abstract Award winner. Floor is yours.
So good afternoon, everybody. Thank you for the opportunity and for the recognition. My poster's abstract number 14, poster's hanging up there. I will be showing a lot of very data-busy slides today that I can't go through in detail, so I'll be happy to explain some of that later on. It's really exciting to be selected as the best poster. It's almost as exciting as Colleen Delaney spelling and pronouncing my name correctly. They said this was easy. Okay, here we go. So I'm a PhD in cell biology. I currently am the director of a cord blood bank. Why would I be talking about clinical outcomes? That is because there's relevance from a manufacturing perspective to look at clinical outcomes in terms of product quality and consistency. Clinical outcomes provide the ultimate measure for safety, potency, but also product consistency. Individual cord blood banks in the US have a responsibility to do outcomes analysis for safety assessment, and we do that based on individual data files that we obtain from the CIBMTR. The CIBMTR has developed a new de-identified cord blood database that includes outcomes for all cord blood banks performed worldwide. So this provides a new opportunity for us and for cord blood banks to look at the impact of product quality and the different types of qualities that have been developed over time. In prior discussions earlier today, and as a theme throughout in the cord blood field, there are in the US 2 clinical types of HPC cord blood product designations. One is IND units. Clinical units need to be IND quality or approved for IND use. Basically, it means these products are manufactured under Factor A/B accredited protocols by accredited organizations. Licensed, or also called BLA units, those are manufactured under FDA-approved procedures as per biologic license approvals. Now, there's always questions around that. Are they the same? Are they similar? They are similar from the perspective of manufacturing. So BLA and accreditation criteria, they can be the same process. It is the acceptance criteria for listing as a licensed unit. They need to pass more stringent acceptance criteria. In the past, in recent years, there were discussions around Zika. So Zika risk, even if the person had a risk but not exposure to Zika, it could not be licensed, it could not be listed as a licensed unit because it would be an ineligible product. Even though it had the same biological properties, the designation did not impact the biological properties. So this is the background for the abstract. We talked about licensed, unlicensed units. But in terms of selection or being part of standardization of cord blood, clinical cord blood transplant processes, one of the parameters is selection of units produced by accredited or licensed cord blood banks. So we wanted to evaluate the premise that US public cord blood banks that have gone through the effort of accreditation and licensure contribute to advancement of clinical outcomes and survival outcomes through delivery of high-quality units that fulfill specific cell criteria, including CD34 and TNC. So the method we used is a retrospective analysis using this de-identified outcomes database. So we can't tell who the recipients or who the donating cord blood banks or who the transplant centers are. That's what we used for analyses. Okay, here. So this is the version that was provided to me in April earlier this year. And with respect to the cord blood banks, this is what you can see there is that as far as the data table told me, 107 cord blood banks delivered just under 15,000 units for cord blood transplants globally. If you look at contribution of individual cord blood banks, you see that 5 cord blood banks each provided more than 1,000 units and accounted for almost half of all those transplants performed. And then the other half is provided by cord blood banks that provided between 100 and 1,000 or 10 and 100 units. So 8 of these 107 cord blood banks have received FDA BLA approval. To date, they have delivered over 1,300 licensed units for transplants, so that's about 8% of that overall. So this gives you a snapshot of the relative distributions. The analysis is broken out in transplants performed between 1988 with the first cord transplant and 2012 when the first licensed product was infused. 3 classes, so pre-licensure IND units, and then after 2012, unlicensed and licensed or BLA products. So this is a busy table, I told you. So what you can see here is that there's a high number of licensed units, information available. And just to give you a quick snapshot, there's a trend for higher use of licensed units in single cord transplants. There was a higher contribution of African American units among the units that were provided, and a low percent of positive results. So great sterility results. And the numbers are very small, the differences are very small, but this is something you would expect from, more stringent and standardized procedure. With respect to product dose levels, on the top there you can see TNC, in the bottom there's CD34. And I just want to highlight, so this is for single cord. If you look at pediatrics and adults there, I guess I could use, here we go. What you can see there is that there is dose levels, but there's also spread in the data. What we find in general for, I lost my pointer here, is that with the licensed units, the spread of the range of the products infused is smaller. So it's more standardized product delivery in that sense. Okay, I should have used my own tools there. So this is where you can see that underlined for the licensed product. The demographics of the recipients that receive these products, some trends that we see there is that there's differences in the average age. So prior to 2012, prior to licensure, it was mainly younger pediatrics. And then see that there's an upgoing trend towards older people in older recipients in licensed units. Again, a slightly higher representation of African American units. And also at the bottom there, a higher percentage of recipients received reduced intensity or non-myeloablative conditioning, different from pre-2012. So here I'll show you some outcomes about engraftment and also at overall survival. And the same thing I want to outline here. If you look at licensed, so this is on the top, neutrophil engraftment for pediatric recipients and adult recipients. And what we can see there is that there is a hint of a slightly faster engraftment for licensed products. But what's more striking is that the range of engraftment is narrower. So they, compared to the unlicensed products in particular, it kind of makes sense. The licensed units are only being produced by eight banks, and also, as said, the release criteria are more stringent. So here is overall survival. This is after single cord transplants in pediatric patients. In black, you see the historical survival rates, and then in red and blue, the survival rates with, for patients that have received either licensed or IND units. And you don't really see a difference there. On the bottom, you can see the numbers at risk over time. There's statistical difference between the historical and then the IND and BLA units used more recently. This is overall survival for single cords in adult recipients, and what we see here is that historically about 30% overall survival rate, and there appears to be a little benefit earlier on between first and year 1 and year 3 after transplant for licensed units in blue there. We have to see how this plays out, but it is an interesting observation. At least it's not worse. So conclusions. So these, what I'm presenting here, are initial analyses. They're aggregate, not segmented for known transplant criteria. So production of increasingly consistent and high-quality cord blood units by public cord blood banks improves to improving clinical outcomes after allo cord blood transplant. And standardization and BLA approvals has helped in supporting improved engraftment and overall survival. So that's evident in the benefit seen if you group licensed and post-2012 IND units together. That is misspelled there, that should be 2012, versus pre-IND, pre-2012 IND units. And as I outlined earlier, licensed unit does seem to provide a tighter spread in the clinical outcomes, which speaks to higher product consistency. So that is something we're particularly happy with to see. It has taken a lot of investment in getting licensure for these 8 organizations, and it's good to see that it contributes to overall improved outcomes. We will still be looking at additional analysis, so frozen product stability outcomes as a function of storage age, CBU age or storage time, and there's information, clinical outcomes available for units that were older than 20 years. I saw that there's a poster from the group of Sergio Quirol as well that speaks to stability there as well, so I'll certainly have a look there. And then we'll be digging deeper into the dose response profiles and sub-analyses where we take these transplant relevant parameters in consideration as well, such as underlying disease, HLA mets, etc. So I want to thank the CIBMTR for making the database available. It's a very valuable research that goes beyond, being able to investigate product quality and consistency, and understand that this is open for application for doing research on that. I think there's four programs that are currently active. I want to thank Joanne. This is not, and Jesse Troy, this is not something I can do by myself. I think it is very important. I think it is very interesting, but I highly appreciate the support that I've gotten there. I work at the Cleveland Cord Blood Center, which is a small independent cord blood bank that would not be able to survive without the dedication of philanthropy that was given by the Katz Foundation and by the Goodman Foundation. And with that, I hope to see you in the poster session, and again, thank you for the reward.
We have We have time for maybe one question if anyone has anyone. Maki, I think this is incredibly encouraging data. Thank you.
I think this is incredibly encouraging data, and I congratulate you for starting this analysis. I do see the differences in the TNCs, and I think that's very well described. However, I would be a little cautious to describe survival advantage based on TNC only. We always want to have multivariate analysis. We have to put in CD34. We also have to put in other improvements in patient care from 2012 and afterwards. So I think this is way more complex, but it's very good that you started it.
Thank you.
Yeah, I think almost most encouraging is that average time to neutrophil engraftment, even taking what Maki just said, right? But how beautiful to see cords engrafting at an average of 16 or 17 days, which I just want to say is equivalent, if not better, to bone marrow grafts, and certainly better than bone marrow with PT-Cy. But thanks.